Hello, this is Ed Chapman, and this is videocast part 3 of photosynthesis. And in this videocast, we're going to focus on the part of photosynthesis referred to as the light reactions. And just as the name implies, this is the part of photosynthesis where light comes into play, and we use the energy of light to move some low energy electrons to higher energy levels so that we can capture them and make some ATP in the process. Now, if you remember from these energy graphs that we've drawn before, reactants that are low down on this graph, okay, because over here this is change in free energy, and this is time, okay. Low down on this graph means low in energy. And the basic problem that photosynthesis has is that it's an endergonic reaction. Energy has to be put into it to build glucose from carbohydrate. And the way this happens is sunlight. Sunlight's going to add energy to the system to raise these reactants to a higher energy level. All right? And so what we're going to end up with is a sugar or a carbohydrate called glucose. And to get there, we've got to perform an endergonic or energy-consuming reaction. So our change in free energy is going to be positive. All right? So this is an uphill push. If you remember, respiration was taking glucose and moving it downhill to release energy in just the opposite direction. Now, this process of making glucose is going to happen in two steps. The first step, which is what this video cast is primarily about, is the energizing of electrons and the making of ATP, referred to as the light reactions, because that's where the energy input is coming from. The second part of photosynthesis that we're going to cover in another video cast is called the Calvin cycle. This is where the electrons that have been energized and captured and the ATP that's been generated are used to build glucose. And another name for the Calvin cycle is carbon fixation. Because if you remember from the carbon cycle, photosynthesis takes CO2 from the air and fixes it into carbohydrate, which is the start of what we call biomass. And the fate of biomass could be the tissues of a plant or moving through a food web, the tissues of an animal, a consumer, or the biomass could get fossilized and become coal or converted to natural gas or petroleum, something like that. So I hope you see the idea of how important carbon fixation is to the sequestration of carbon in a place that's not in the atmosphere. All right. Now the light reactions do four things. All right. The light reactions extract electrons from water. Now these water electrons have very, very low energy. All right. So they need to be literally bounced to a higher energy level. All right. And this energy is going to be coming from sunlight. And then these electrons are going to be allowed to in a way, fall back downhill. While they fall, this energy is going to be captured to make ATP. And of course, the process for doing this is chemiosmosis, exactly like we saw in mitochondria using the enzyme ATP synthase. All right, so let's focus in on the light reactions now. If you remember, I said that water is a very low energy molecule. It's oxygen bound to two hydrogens, and because oxygen is so electronegative, all right, oxygen, of course, is water is HOH, and it's got two polar covalent bonds here. The electrons that make up these bonds are being controlled by the oxygen because oxygen is so electronegative compared to hydrogen. So there is very little energy in these electrons. So in order to energize these electrons, we've got to move them uphill, all right? And in the process of doing that, we're going to take them away from oxygen. So oxygen, in this case, is being oxidized. All right. Now think about how weird that sounds. Oxidizing oxygen. Okay. Oxygen is going to be losing electrons. Remember, oxygen in respiration is getting electrons. So in order to take electrons away from oxygen, we're going to have to really use a lot of energy to make this happen because oxygen is not going to give them up without a fight, so to speak. So the whole purpose of the light reactions is to move these electrons to a higher energy level, and in the process create some hydrogen ions. And the leftover oxygen, which has had its electrons taken away, combines with another oxygen to produce oxygen gas, which of course is one of the products of photosynthesis. All right, so let's look at this idea of electron excitation, or exciting electrons. All right, now the key to all this is a pigment molecule called chlorophyll. All right. Now, chlorophyll has a really interesting property. Because of its structure, its molecular structure, chlorophyll, if you shine sunlight on it or shine light on it, it will kick an electron out and this electron will rise to a higher energy level and then fall back down into the chlorophyll, into the exact same place it came from. And as the electron falls back downhill, it's going to release the energy that it once had. And this energy is going to be released as heat and light. It's going to release a photon of light. So in this beaker right here, we have some green chlorophyll that's been illuminated with really strong light, and then the lights are turned off, and the chlorophyll will actually glow a reddish color for a few seconds. And this is because it's fluorescing. All right, it's a fluorescent molecule. This is proof positive that the chlorophyll is releasing the energy as light. All right. Now, the reason why the light is red coming out is because it's a longer wavelength. It's lost some of its energy. It's coming out at a longer wavelength than it went in. And of course, that's because of the second law. Some of the energy is having to be taken away from it and lost as heat. Now, photosynthesis doesn't do this. Okay, if you think about it, the electron is being bounced up and then it's falling right back where it came from. This is a loop that doesn't make anything. We're not getting any ATP from this. But what if, while this electron was up at this higher energy level, we allowed it to fall back down an electron transport chain and use that energy to turn ADP into ATP? All right? That is what chemiosmosis does, and that's exactly what the light reactions are going to use to make some ATP. So let me show you what I'm talking about. All right? Green plants build their chlorophyll molecules and other pigment molecules into something called photosystems. And photosystems capture photons of light shining on them and energize electrons, just like we saw. But these excited electrons are passed from receptor to receptor within the photosystem until finally they gain enough energy to be accepted by something called the primary electron acceptor in an area called the reaction center. All right, so all of these pigment molecules around the reaction center are just there to capture photons of light of different wavelengths, okay, and bounce them around and add energy to them with each bounce until finally they are energetic enough to be captured by an electron acceptor and be passed through an electron transport chain. All right, so photosystems, there are two photosystems. Photosystem 1 and Photosystem 2, and they work together to shuttle these excited electrons to an electron carrier, just like we saw in respiration. Except the electron carrier in photosynthesis is slightly different. It's called NADP+. And when NADP+ captures electrons, it changes into NADPH, which is, of course, the reduced form. And chemiosmosis is then used to make ATP from the energy of these energized electrons. All right, now this diagram looks complicated, but I'm going to break it down for you. All right, here we have photosystem 1 over here with all the antennae, pigments, and the chlorophyll at reaction center right here labeled P700. And over here we have photosystem 2, a similar situation. It's got a chlorophyll molecule at its reaction center that's called P680. Now these numbers are light wavelengths, and so P700 is a little bit longer and a little bit redder light, and P680 is a little bit shorter and a little bit bluer light, if you remember the electromagnetic spectrum we talked about back in part 1. And we're going to start with photosystem 2. So what photosystem 2 does is it gets struck by light. Okay, the light energizes electrons. They bounce all around in the photosystem here, and they finally get bounced to a high enough energy level where they can be captured by a primary acceptor. All right, then they pass through an electron transport chain, falling downhill, and finally giving their energy up enough to reduce NADP+ to NADPH. So the product of photosystem 1 is NADPH. So we have literally moved an electron from chlorophyll through an electron transport chain to NADPH, or a pair of electrons actually. All right. Now, if you think about it, if this just ran by itself, they're going to run out of electrons down here. So they've got to be replaced. So jump over here to photosystem 2. Same situation. Light is shining on photosystem 2 in the membrane of the thylakoid, and it's energizing an electron to a higher energy level. It's captured by a primary acceptor, but this time, as the electron falls down an electron transport chain, or what they call a cytochrome complex, it's actually used to do chemiosmosis and produce ATP. All right, so we're getting some ATP made here. But the beauty of it is that electron then falls back down into photosystem 1 and replaces the electron that was lost in this pathway. Now, if we go over here, if we lose an electron here, something's got to replace it in photosystem 2. And that replacement is coming from water. Okay, so water is being broken up into hydrogen ions, oxygen, and electrons. And these electrons are then feeding into photosystem 2 to actually be the source of electrons. So putting the whole thing together, if we start over here, electrons from water into photosystem 2, bounce to a higher energy level, a trickle down an electron transport chain, making some ATP while they fall, replacing the electron here, which then gets bounced up, trickling down a little ETC pathway here to finally end up in NADPH. Okay, so the products of the light reactions are twofold. We have NADPH and we have ATP, two forms of energy. And that's what the light reactions do. Here's another way of looking at it. I kind of like this one because it involves little people moving around. You can think of this as photosystem 2, and this is photosystem 1, just like we saw before. But here they're using the idea of kinetic energy. So light energy is, in a way, kind of like somebody hitting a fulcrum point here on a lever. What's that called again? Oh man, what's that called? A lever, I guess. So hitting this fulcrum, it bounces the electron up. It's captured by a primary electron acceptor. It's rolled down a hill, which involves a kind of turning mill here. Okay, a lot like that ATP synthase. And it's used to pump hydrogen ions across a membrane, and then ATP synthase allows them to flow back, and we get ATP being produced. Then the electron hits another little fulcrum point here in lever, where it gets hit again by another photon of light, bounced to an even higher energy level, and finally added to NADP plus to make NADPH. All right, so I hope between those two different diagrams you can see kind of the chain reaction combination of situations here that are allowing light energy to be converted to the chemical energy of ATP and NADPH, which is going to be critical for the Calvin cycle, which is what's coming up next. All right, now if you just ran photosystem 1 by itself, okay, you get something called cyclic electron flow. So if we just completely left out photosystem 2 and just ran photosystem 1, where you bounce electrons up, run them through an electron transport chain, and make your ATP, okay, just like we did before, but then shuttle that electron right back to where it came from, we get ATP out of this process, but we don't get any NADPH. Okay, so cyclic electron flow is just photosystem 1 running all by itself. And plants certainly can do this if they just need to make ATP for respiration needs and all that stuff. All right, NADPH is a product that you do not get out of cyclic electron flow. All right, we'll stop there and pick up with part 4, looking at chloroplast structure and how chemiosmosis works. Thanks for listening.